All right, so now we're going to take a look at our primary lymphoid organs and some lymphocyte development, and then we'll take a look at our secondary lymphoid organs. So our primary lymphoid organs are the bone marrow and the thymus gland. So these are our primary, meaning our initial site of making some of these lymphocytes, where they start from. So our bone marrow, we talked about already in the blood and cardiovascular system, mostly the blood system. The red bone marrow is a loose collection of cells where hematopoiesis occurs, and the yellow marrow is a site of energy storage, largely fat cells. So for this purpose, for our lymphatic system and immune system, we're more concerned with the bone marrow, which is the red bone marrow, where we have hematopoiesis. This is where our B cell undergoes nearly all of its development. Remember, in the red bone marrow, B for bone marrow for the B cell. And then our immature T cell is made here. It's called a thymocyte. And then it leaves the bone marrow and then matures in the thymus gland. So then when we talk about our other primary lymphoid organ, we're talking about the thymus gland. So the thymus gland is a bilobed organ found in the space between the sternum and the aorta of the heart. You probably already looked at it when we talked about the endocrine system. The connective tissue holds the lobes close together, but it separates them as well and makes them a capsule right in this region here in the chest. And then, of course, this is where we have our thymus maturing. So it divides the thymus up. The cortex is densely packed, so it stains darker. You can see slides of that. But then the medulla is where the thymocytes migrate before leaving the thymus, which contains a less dense collection of thymocytes, epithelial cells, and dendritic cells. So what we see is that we have our two primary lymphatic organs, which are the bone marrow and the thymus, because our bone marrow is where we have all of our lymphocyte production to start with, and then our B cells are going to mature in the bone marrow, and then our T cells mature in the thymus. So when we take a look at our secondary lymphoid organs, we first want to talk about the naive lymphocyte. So that's one that has left the primary organ, so it's left either the bone marrow or it's left the thymus, and then it has now entered a secondary lymphoid organ. So a naive lymphocyte is just one that hasn't entered its secondary lymphoid organ and hasn't differentiated and specialized at that point. They are fully functionally, immunologically, but they haven't encountered an antigen to then respond to yet. This information I'm not going to have you be responsible for. Those are some of the things that all of these tissues have in common, but we're going to go through each of these secondary lymphatic tissues, or lymphatic sites here, our lymphatic organs. Let me be more clear. So the first one that we're going to look at are our lymph nodes. So the lymph nodes remove debris and pathogens from the lymph. We've already talked a bit about these. There are hundreds found all over the body, especially along our major blood vessels. They filter the lymph. So we have our lymph going into our lymph nodes, and then it gets filtered within the lymph node, and then it leaves the lymph node and continues up until we get to the subclavian veins again. So we have our any bacteria that are going to be taken up. Anything that we don't want is going to then go into a lymph node at some point. We get it from, say, in the body, it's in the tissue, then it's going to get pushed into the lymph node, into the lymph capillaries, and then travel up into a lymph node. We have macrophages in our lymph node. We also have T cells and B cells. So what we see here is that the structure of our lymph node, you can see here's a slice of it here in actual tissue, and then an illustration here. We have our fibrous capsule that's on the outside, our connective tissue, our fibrous capsule that's going to be holding it in place. It surrounds the node, it separates it from other tissue, divides it into segments. And so what we see is, if we look at the illustration, you can see this is our fibrous capsule on the outside here. And then if you look maybe over here, you can see that it folds in and divides it into sections, into interior segments. Then within this interior portion, we have the cortex, which is this outer region here. You can also see the cortex labeled here, this darker staining region. and that's going to contain lymphocytes for defense. So it's going to contain B cells and T cells. And then we have our medulla, which is our inner region here. And you can see in here, where it's a little bit lighter stained, is where we have our medulla region. And then the medulla region is going to contain macrophages to phagocytize debris and bacteria and viruses and cells and things like that. So what we see here is that we have our afferent lymphatic vessels, which is where we are going to have our lymph flowing into. And we have many afferent lymphatic vessels, all coming from our various collecting vessels, which came from our capillaries throughout the body. Afferent, the way I remember it, A comes before E, which is efferent lymphatic vessels, which is where it exits. E is exits, and A is where it comes in at the beginning. We have many afferent lymphatic vessels, okay, which is going to carry the lymph into the lymph node. And then we have 1 to 3 vessels that are the efferent lymphatic vessels, where we have it leaving. And what we see there then is that we have more fluid coming in than we have leaving at a time. And so we have this kind of bottleneck where we bring fluid in and it has to stay in there for a while, so that the B cells and the T cells that are in the cortex can do their work and try to get rid of any invading pathogen and debris. And then also the macrophages in the middle section here in the medulla will be working on it to phagocytize things. We also, in the medulla region, we have our plasma cells, and we have some other B cells. And so we kind of have a lot of fluid coming in, it bottlenecks in there, it's stuck in there, flowing around, which gives all of those cells time to go through and filter things out and kill things before then it leaves via the efferent lymphatic vessels. Then that's going to carry it up, and then the lymph nodes, since we have many of them throughout the body, are going to carry the lymph from lymph node to lymph node to lymph node. And then as it's going through, it's going to continue in this bottleneck fashion and give that time to be filtered out, so that once we get up to our subclavian veins, we have clean lymph that's going into our bloodstream and not causing any issues further. So the function of our lymph nodes, here are: 1 is to filter the lymph and remove bacteria. Okay, so remove anything that's in there, bacteria, viruses, cancer cells, dead cells, debris. And then also it's the site of B and T lymphocyte activation for an immune response. So we have a lot of these B cells and T cells in there. And then when the lymph comes in there and it encounters a pathogen, then we're going to have activation of these B cells and these T cells. And so what we see, again, remember the B cells are going to activate and then turn into plasma cells. And then we're going to have the plasma cells send out their antibodies, seeking out the antigens and telling everybody else, hey, this is a bad guy, let's get him. Same thing with our macrophages. Our macrophages are going to send out the message saying, hey, I've gobbled this guy up, look for more of this guy. So that's our second function, is that activation of the immune response. And then the first function is to just do that function, is to get rid of the things in there. All right, so next we're going to take a look at the spleen. So another secondary lymphatic organ is the spleen. And we talked about the spleen already when we talked about blood, but it's also a secondary organ. Its function is mostly here to produce blood in the fetus. It's a very fragile organ, as you can see here. It's sometimes called the filter of the blood because of its extensive vascularization. It has macrophages and dendritic cells that remove the microbes and other materials from the blood. So it is part of that lymphatic and immune system. So the first function of the spleen, which goes back to the blood chapter, is that it produces blood in the fetus and also in anemic adults. Remember, we have the ability to kind of kick our spleen into gear when we have anemia, and it'll start working for us. Secondly, it removes old blood cells from circulation. We talked about that in the blood chapter as well. And then 3 is the function that's related to this chapter specifically, in that it filters the blood and then also activates the immune system. So since we have these macrophages, again, they activate the immune system in sending out messages saying, hey, we need to get rid of these guys. I've encountered this bad guy. So It also functions as the location of the immune responses and blood-borne pathogens, so something in the blood. So the liver and the bone marrow make and remove red blood cells if the spleen is removed. But if the spleen is removed, there could be an increased risk of infection because of this immune response that it has as well. So the structure of the spleen, you can see here, we have this fibrous capsule. So we have, it's about, let's look up here, about 12 centimeters or 5 inches long, relatively small, and is attached to the lateral border of the stomach via the gastrosplenic ligament. It's fragile, without a strong capsule, and is dark red due to extensive vascularization. So it doesn't have a really strong capsule. It's very fragile. It breaks very easily, especially in surgery situations. Surgeons have to be very careful of the spleen because they can damage it very easily. So we have this, what is called red pulp and white pulp. So the red pulp has, so you can see here's the white pulp that's labeled, and then the red pulp here. Red pulp there. So the white pulp, as it is colored here as white pulp, contains lymphocytes and macrophages. So you can see we have our blood flow here. The blood can flow in, and then right next to our blood flow there in the arterial, we have our white pulp that contains lymphocytes and macrophages that are going to be filtering the blood, or the fluid coming out of the blood here. And then we have our red pulp, which is filled with our sinusoid capillaries. Remember when we talked about our cardiovascular system, we saw sinusoid capillaries in our spleen. And so then our red pulp contains those sinusoid capillaries that are filled with red blood cells. So we have this region where we have, remember the sinusoid capillaries were very leaky, kind of leaky spaces of blood. So we have all of these leaky spaces of blood here that are leaking fluid around, leaking blood around, and then we have our white pulp within there that has our immune system function, our lymphatic function. All right, so then we are going to move into our lymphoid nodules. So if you remember back to our levels of organization, when we have a bunch of cells together, we have a tissue. And so lymphoid nodules are kind of lymph tissue that isn't specific to a lymph organ, but they are sometimes found in lymph organs. So sometimes they can be temporary when we have a bunch of these lymph cells, so the immune system type of cells, all coming together in an area. So if there's an infection, a large mass of bacteria that got into the system, we have a lot of this, like an aggregation of these cells all coming together. And then when that happens, we have what's called a lymphoid nodule. So they have a simpler architecture than the spleen and the lymph nodes because they're not a primary lymph organ, but they're a dense cluster of lymphocytes without a fibrous capsule surrounding it. That's why it can be a temporary situation, because we see all of this aggregation, this cluster, and then it can then go away once the bacteria is gotten rid of or other pathogen. We do see these nodules in the respiratory system and digestive tracts, areas routinely exposed to environmental pathogens. So things that are open to the exteriors where we see these aggregate. We do see them, these masses of lymphatic tissue, found in many of the lymphatic organs. So for example, tonsils are another lymphatic organ. All right, so the tonsils then are going to be lymphoid nodules that do contain a capsule, some sort of casing that's going to separate them from the tissues around them. Rather than being an aggregate of cells that can come and go, then we have these lymphoid nodules contained inside tonsils. Let's scroll down just a minute and look at the image here so we can see where the lymphoid, or where the tonsils are located. We have 3 sets of tonsils where we have our nasal and oral cavities. So you can see up at the top region here, if we look at this image here, so this is our nasal cavity, we breathe in here. So at the back of the nasal cavity, we have 1 set of tonsils, and these are our pharyngeal tonsils, sometimes called the adenoids. And then we have a second set back over here, which are called the palatine tonsils, back here at the back of the oral cavity. And then it's in a blown up picture down here, so back near the throat. And then we have our third set of tonsils, which are the lingual tonsils, which lingual is related to the tongue, and they're underneath the tongue here. And you can see back here also, this is the tongue here, so they're located at the base of the tongue. So if we go back up, we can talk about these tonsils and what their function is. So unlike a lot of the other organs that are lymphatic, the tonsils are not actually meant to filter through things. They're not like the lymph nodes where they go through and they filter things and clean up lymph. The tonsils actually are kind of taking a taste of things as they come in and then sending messages out to the rest of the body to mount an immune response. So we have these tonsils located in the various regions. Again, the pharyngeal tonsil are also called the adenoids sometimes, specifically when swollen. And then swelling is an indication of active immune response to infection. So what we see, I'm actually going to scroll down to the image here again, because if we look at the histology of the palatine tonsil, we can kind of get a better picture of what it is that we're looking at in the function of the tonsils. So if we look at our palatine tonsil here, if we take a slice of it, what we see are these crypts, which are an infolding or a pit, basically, that goes into the tonsil. And this pit is lined with lymphatic tissue, so lined with those lymphatic nodules. And what we see happening is, remember, they're at the back of the nasal cavity, the back of the throat or the oral cavity, and then under the tongue. So what happens is when we're eating or drinking or breathing in, everything that we're eating, drinking, breathing in is going to pass over these tonsils and then get some of that into these crypts. Once that gets into the crypt, then this lymphatic tissue in here is going to go through that and kind of check out what's in it. So if it finds out that there's some bacteria in it or virus in it, then it can mount an immune response. So within the tonsil itself, we'll see the tonsil kind of killing off those things as they're stuck in the crypts. But then at the same time, we have the messages being sent out to the rest of the body saying, hey, incoming, they're these guys, they're coming in, we just caught them. And so then it's going to get the rest of the immune system working in that same way. So anything that is inhaled or anything that is ingested, it's going to then activate the immune system. That's the function of these tonsils, is to kind of take a taste and then send out information to the rest of the system saying, hey, this is coming in. So the tonsils themselves don't act kind of like the lymph nodes where they're actually filtering through things. It just takes a little bit and then sends that message out. And so if we do look at this histology here, you can see the crypts. And then on the outside, we do have this epithelial tissue and a capsule region. And then we have germinal centers. So we do have all of these lymphoid centers, these clusters of tissue, these clusters, these lymphatic nodules, clusters of tissue that are going to function in that way. They're going to not only kill the stuff that comes into the crypts, but they're also going to send messages out saying, hey, these guys are coming in, let's mount an immune response and get rid of it as soon as possible. So the major function of the tonsils then, specifically we see them, they're larger in children, but it's a good way in children's bodies to recognize foreign invaders, foreign pathogens, and then destroy them because they're stuck in the crypts there and they're going to go through and destroy them. But then it also is important for developing immunity to common environmental pathogens so that they're going to be protected in their later lives. So the main function there is to see what's coming in, send out messages, and in a lot of cases, we have these long-term or memory cells that we'll talk about more in the immune system that are going to then maintain that immunity throughout the life. So it's very important. It does mention here that tonsils are often removed in children who have recurring throat infections, specifically or especially those involving the palatine tonsils on the other side of the throat, because it may interfere with breathing and or swallowing. So it is a really difficult decision because if you take those out, then the body is going to have a less of an opportunity for it to pick up incoming pathogens and then develop a better immune system later in life. But then, of course, if it's going to be a problem with breathing or swallowing, that also is not good for life. So that is something that needs to be taken into consideration. So now we're going to take a closer look at two other types of lymphatic tissue. So when we were talking about the specific organs, we talked about the lymph nodes, we talked about the thymus, we talked about our spleen, and then we talked about lymphatic tissue, as in we talked about the lymphatic nodules, which was just a mass or a dense aggregation of cells that can come and go. We also see two other types of lymphatic tissues, or there are a couple of other types, so we're going to talk about two of them in the text. So the first one out of these three, so lymphatic nodules, then MALT, is mucosa-associated lymphoid tissue. So here, mucosa-associated lymphoid tissue, or MALT. So this is an aggregate of lymphoid follicles, so kind of this, again, aggregation of cells, but these are directly associated with mucous membrane epithelia. So we're talking about mucous membrane areas. So we're talking about things in the digestive system, for example. So here, in the underlying mucosa of the gastrointestinal tract, so the digestive system, breast tissue, in the lungs, and in the eyes. We see Peyer's patches, which are a specific type of MALT, the mucosa-associated lymphoid tissue, in the small intestine. And these are important in the immune response against ingested substances. So as we're eating food and it's being broken down in our small intestine, these Peyer patches contain specialized endothelial cells that sample the material from the intestinal lumen, from that hole in the intestine, the middle portion, where everything's being broken down, and then it's going to transport it to the nearby follicles so that we can start mounting this adaptive immune response, this longer, slower term immune response, so that we can get rid of those pathogens. So we see Peyer's patches in the small intestine, but we see MALT, which is the larger grouping here. Peyer's patches is a type of MALT. We see MALT in various areas, including breast tissue, lungs, and eyes. And you can see here, they have a histology down here. You can see the mucosa of the intestine here. This is our lining here of the intestine, and we can see this is the lumen area here. And then you can see the Peyer's patches are these lumps of lymphoid tissue surrounding or within the outer region of our small intestine, so that they're just taking little tests, little samples of the food that we're eating and the liquids that we're drinking to, again, mount an immune response. And then the last of the lymphoid tissue that we'll talk about is BALT. So BALT is bronchus-associated lymphoid tissue. So again, it consists of lymphoid follicular structures. So we're talking about, similar to lymphatic nodules, clusters of cells that are coming together. But in this case, they're not transient. They don't fall apart when there isn't anything to fight. But these are found in the bronchi, these bifurcations of the bronchi. A bifurcation is where it branches into two parts. So when we get into the respiratory system, we'll see these again. But we see these lymphoid follicular structures, and then they have this overlying epithelial layer, so kind of a capsule layer, and they're found along the bronchi. And the bronchi are basically our breathing tubes within our lungs. So when we breathe in air, it's going to go down and then get into our lungs. And in that region, we have this kind of tree of tubes that includes bronchi. And these bronchi then have this tissue around it, so that whenever we're breathing in, similar to the MALT in the Peyer's patches in the small intestine, where whatever we're eating and drinking, it's going to filter that out. It's going to take a little sample and send out messages. In the bronchus-associated lymphoid tissue, the BALT, we see that same thing. Whatever we're breathing in, if we start to breathe in some bacteria, we have this tissue in our lungs that's going to sample that, say, oh, there's some bacteria in here, and start sending out this immune response. All right, so that is the end of our actual lymphoid tissue and the lymph system. And then starting with 21.2 and moving on, we're going to talk about more specifically the immune system and take a closer look at some of the immune system cells, the lymphocytes and leukocytes.